Hello children. So today we are going to learn about the sexual reproduction in a plant. So before going to discuss about the reproduction in plant, first let's see what is a flower. So do you know what is a flower? A flower is called as the reproductive organ of a plant. So you have learned about flower in detail in the junior classes also. Let's a quick recap about the flower once again. So what is a flower? We are saying that flower is a reproductive part of a plant. So why it's called like that? First see that in how many holes the flower is divided. Holes means in Hindi you can say chakra. So how many steps the flowers is divided. So flower is divided in two holes, that is the accessory hole and second one is the essential hole. So see here, what are the accessory holes? The accessory hole first is the calyx. This green portion, the outermost portion of a flower is called the calyx, and the unit of calyx is called sepal. So what are the accessory hole? First is the calyx, and the second hole of a flower that is mainly colorful in nature. So that is called the corolla. This is called the corolla. This is called the corolla, and the unit of corolla is petal. The unit of corolla is petal and the unit of calyx is called sepal. So mainly why these are called accessory hole? Because they are just to decorate the flower, to in case to attract insects towards it. So these flowers are called as the accessory hole of a flower. Now see what are the essential hole. Without that, the flowers can't do anything. So these parts of a flower are called as the essential hole of a flower. So for that, what's happening? That here the essential holes are the sex organ of a flower. These are the androecium. Androecium is the male part of a flower and the gynoecium. And what is and the gynoecium? So gynoecium is the female part of a flower. So see here, anther. This is the male part of a flower, and this is called the gynoecium. This is the female part of a flower. Now see the gynoecium is again divided into how many parts? It's the upper part of a gynoecium is stigma, the middle part of a gynoecium is called style, and the lowermost part of a gynoecium is called as ovary. Here you can see the androecium, it's a anther-like anther. This is anther which is mainly in bilobed in nature. So why it's called as the essential hole of a flower? Because this gynoecium and androecium, the female gamete and the male gamete, they both take part in reproduction, due to which you can see the formation of seeds, the formation of fruits happened. That's why these flowers is called as the reproductive part of a organ. Just note down this. Now here you can see that we have made diagram of the androecium, that is the stamen. Stamen is the male part of a flower. So as we discussed that about the flower structure, and we come to know about the sex organs of a flower, that is male gamete and female gamete. And here this is what the picture of a stamen, that is the anther. What is the anther diagram? So this is the male sex organ of a flower. Now what you will see here, that these are the two structure of a stamen, and what is the structure of a stamen? It has the long and slender stalk. So this is the long and slender stalk, that is called as the filament. What is this called? This is called as the filament, and terminally generally bilobed structure. See here, it's a bilobed. Bilobed, why? Because it's in two lobes. Bi means what? Bi means two. So it's in two lobed. That's why it's called bilobed structure. It's called what? This bilobed structure is called the anther. This bilobed structure is called what? It's called as anther. The proximal end of the anther is mainly attached with what? With thalamus or the petal of a flower. The proximal end of a anther is mainly attached with the end of the petal or the thalamus of a flower. And you will be very surprised that the number of the anther is variable in different flowers. It's maybe the 2, 4, 5, 6, 8, whatever, and the size is also different in all the flowers. It's not always remain same. And if you want to see this, you can easily see this on the arranging the anthers of a flower on the slide, and you can see, you will be able to see under the microscope that how these are the variation of anthers in the flowers. Now, a typical angiosperm anther is what? It's always bilobed, with each lobe having two theca. Now see here, it has two theca. This is one theca and this one is second theca, first and second theca. So mainly a typical angiosperm anther is bilobed with two theca, having two lobes and having two theca. So they are called what? They are called what? Dithecus. They are called what? Dithecus means, di means two, thecus means theca. That's why they are called what? Dithecus. So often longitudinal groove runs lengthwise separating the theca. See, you can see here longitudinal groove running here, and which divide these two theca from each other. Okay. Now let us understand the various types of tissues and their organization in the transverse section of an anther. So what we are learning, it's a transverse section. So we are showing here the typical structure of a flower. Now we will see the transverse section of a anther, that exactly what is the structure of the anther, how the spores come out, how the pollen sacs come out. Pollens are coming from the anther. So you can see here, these are the pollen grains. These are what? These are called pollen sacs, and these red what you can see is pollen grains. This is what? Line of dehiscence. So what is line of dehiscence? Means on maturation of the anther, this burst out. From here, the anther burst out and all the pollen grains come out from this anther. So this is called, that's why line of dehiscence. Now let us, you just note down these things, and we will learn about more about the anther, that how it's functioning and what are the main structures. Now let us see the structure of a transverse section of a anther. So earlier we have seen that what is the structure of a anther. Now see the transverse section of a structure of the anther. So here what's happening, that we know that angiosperm, this anther is a bilobe. After that was the anther is a four-sided structure consisting of four microsporangia located at the corners. So see here, the anther is a tetrad. 1, 2, 3, 4. So it's microsporangia consisting of four microsporangia. So here is four microsporangia located at the corners and two in each lobe. So this is one lobe and this is second lobe. Now the microsporangia develop further and become what? Pollen sac. This will again become mature and that will become what? Pollen sacs. They extend longitudinally. So earlier what you said, that they extend longitudinally, the length and an anther and are packed with pollen grains. Now let's see the structure of microsporangia. So these are the microsporangia. Now see the structure of a microsporangia. So in this transverse section, what you can see that the it is generally surrounded by 4 wall layers. So this microsporangia is what generally covered with 4 walls. So what is these walls? First is epidermis, second endodermis, third one is middle layer, and fourth one is what? It's a tapetum. It's what? It's a tapetum. Now the outer 3 wall layers, what they are doing, their function is to protect the spores. So what is the function of these three? Epidermis, endodermis and middle wall is to give protection to the microsporangia. Now what the tapetum is doing? So the innermost wall. So what is tapetum? It's a innermost wall, and it's giving what? It's nourishes the developing pollen grains. So what is the function of tapetum? It nourishes the developing pollen grains. Now see the cells of the tapetum possesses dense cytoplasm. It consists of dense cytoplasm, and it's generally have more than one nucleus. What tapetum has? It's generally have more than one nucleus. Now see what's happening. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue. What's called the sporogenous tissue? So what is sporogenous tissue? It's the group of compactly packed arranged homogeneous cells. Okay, so this homogeneous cells, group of homogeneous cell is called what? Homogeneous tissue. Yeah, so this is called as the homogeneous tissue, which is called what? Sporogenous tissue. This homogeneous tissue is called what? Sporogenous tissue that occupies the center of each microspore. So what it occupies? The center, see, the center of each microspore. Now let us see the formation of microspores. So how this microspore formation happen and what this function process is called as. Yes. So here for the formation development of the anther. So what's happening? The anther develops, the cells of the sporogenous tissue undergoes what? Meiotic division. What it's going? It's going in meiotic division. Yes, so what's happening? Meiotic division for this. So microspores go under meiotic division to form what? Microspore tetrad. What the anther is doing after on development, what they do? The sporogenous tissue, what they did? They are going under meiotic division to form what? Microspore tetrad. So now say when they are going under meiotic division, so microspore tetrad, so what will be their ploidy? What will be their ploidy? Means they will be all N. They will be all what? N. Yes. So that when this N and N means these gametes will again take part in reproduction, they will become what? 2N. So now here after meiotic division, what? These ploidies are N. These are what? These are N. Now you can see that at each cell of the sporogenous tissue is capable of giving rise to a microspore tetra. So each cell is capable of giving rise to a microspore tetrad. Yes. Now the each one is potential pollen. What? Each one is a potential pollen. You can see here the extended structure: epidermis, endothecium, middle layer, microspore mother cell, and tapetum. So you can see here microspore mother cell. This is the enlarged structure of this. So what's happening? Each one is a potential pollen or microspore mother cell. So here what is? These sporogenous tissues, these are capable of giving rise to a microspore tetra, and these each is a potential pollen or microspore mother cell. That is what? microspore mother cell, MMC. Now the process of formation of microspores. The process of formation of microspores from pollen mother cell through meiosis. The process of formation of microspore from pollen grain, pollen mother cells. So the process of formation of microspores from a pollen mother cell, that is what? PMC, pollen mother cell, through meiosis. Here we see through meiosis is called what? Microsporogenesis. Is called what? Microsporogenesis.